Let's get to the highlight. CPE, the three letters, citopatic effect, the control experiment. Oh yes, you were gonna show us a little bit about that. Indeed, I now have the first results from the control experiment. They are here now, and they clearly show that the effect that all virologists take as a proof that the tissue or the cell material turns into virus, we can perform the same exact results with the methods, only without allegedly infected material added. We have here the pictures that we can fade in. Can you do that, or can I do that? I think you should be able to do it now. I gave you access to it. Here we go. Yes, those are the control experiments. These are the freshly isolated tissues that are separated, then in this form referred to as a cell.
Bezeichnet werden.
And here we have converted them to the medium that virologists use. We have added 10% fetal bovine serum. We took low amounts of antibiotics so they can get used to it. And here it's actually when the experiment starts. We reduce, like all the virologists do, nutrient solution from 10%, which is normal, drastically down to 1%. Then add huge amounts, 3 times antibiotics. And we can already see that the cells and the tissue don't like that. Already 1 day after the transfer. And especially after 5 days we can see massive decay, and exactly so specific and in the same manner like virologists interpret that. This is the cell toxin, the cell killing, the cytopathic effect, abbreviated as CPE, and we can see this even more clearly when I added another source of RNA. This one comes from yeast, not standardized yeast, but messenger substance. The RNA taken from yeast to verify, to see how the tissues, the cells deal with it, as nothing other is being made when I add infected material to the cells, because that's full of nucleic acid, and the decay and the death of the cells is even more dramatically. The day before yesterday I have already received the results from the RNA that we isolated from it, from the controls that have not been treated, from the differently treated virum probes. We have isolated the RNA. This is now in the sequencing, and the quality of the RNA is extraordinary for our purpose. I received the results yesterday. Thereby, virology is disproved in the first step, because we clearly demonstrate that the central effect, the dying of cells in the test tube, we can create in the same manner without infected material. And virologists should have made such a control experiment since 1954. That until today it is not documented that they did it. So, and this equating with this effect, cytopathic effect, CPE, and this is for the virologists the proof of a virus, and then they're calling their dying cell tissues an isolate, which then they offer on the market for 2,000 euros, and then they say, and in here, and this is the virus, and from that we can create a vaccine now. We can take the whole and then we call it a live vaccine, or they take out individual proteins and call it an inactivated vaccine, because there's only a component of the virus present and that makes it not infectious. And this is claimed as an inactivated vaccine. And this is what we have achieved. This is a third of the refutation. These tissues now are in the electron microscopy, and I can already say they will detect the same exact particles in the non-infected tissues that virologists always claim to demonstrate as virus particles. This is happening now, and the pivotal in the virology with the nucleic acid, it's being sequenced now, and then we do just like the virologists do. We construct with their own programs that are already running on our computers. We construct the same exact genome as SARS-CoV-2, HIV, measles, or Ebola from exact this material that was never infected, that has never been in contact with material from a patient. These are the control experiments, and part 1 is successfully completed, and now we're waiting for the electron microscopy and then later for the genetics. But here the central argument of the virology has been refuted. This cell effect, this citopathic effect, that tissue cells die, that this is a virus, we have now thoroughly disproved. This is the first success, and here I present this data for the first time publicly. This is a small sensation, or a huge sensation, but you already knew it beforehand. Yeah, I knew it, and as a biologist I know it. That it comes like this. We already had it with the measles. With the measles virus I did it just like that. Back then the RNA was not sequenced to disprove that. We didn't need it back then.